Joydeep is going to tell about the story of TMEM16F on exocrine pancreatic physiology.
So today I am going to talk about the phosphatidylserine scramblase TMEM16F, or also known as ANO6, and its role in exocrine pancreatic physiology. So this entire work was done during my postdoctoral training at NIH, Dr. Smul Willem, and I recently joined CU Raj around 3 months ago. So before going to the data, I like to remind how the pancreatic physiology works. As all of you know, the exocrine pancreas secretes the hormones, whereas the endocrine pancreas, whereas the exocrine pancreas secrete digestive enzymes and the fluid. In response to calcium elevating agonists that stimulate the secretion of the digestive enzymes, as well as calcium stimulate the ANO6 channel, chloride channel that stimulate chloride secretion. Whereas increased cyclic MP activated by the BIP may activate chloride channel, but the molecular identity is not known. Is this ANO6? So when the chloride, fluid pass through the ductal cells, it will get modified, the chloride reabsorbed and bicarbonate secrete. So bicarbonate rich fluid come out from the pancreas. So anoctamin-6 is a family member of calcium activated chloride channel that have other 10 family member. ANO1 is a bona fide calcium activated chloride channel but has no PtdSer Scramblase activity, role in the epithelial secretion and the muscle contraction. ANO8 is important for the threader protein and calcium signal regulation. Other function as a chloride channel as well as lipid scramblase like ANO6. You can see it can bind with the calcium, also as well as that I found during my PhD training, it can bind with the membrane PIP2 and regulate this channel function. So let's see how ANO6 function as a PtdSer scramblase. So as you know, the phosphatidylserine present in the inner leaflet of the membrane in the resting condition. So when the get activated by the calcium, it transports the PS from inner leaflet to outer leaflet. That's why it's known as calcium activated PtdSer scramblase that have several physiological functions like blood coagulation, apoptosis, mineralization, or cell-cell fusion event like fertilization, or so on, or membrane trafficking that we are going to discuss today. So till now date, there is a one ANO6 mutation loss of function that known as mild blood coagulation disorder, Scott syndrome. So it also have the chloride channel activity that I first mentioned for the physiological relevance, that it get activated by the cholera toxin and leads to secondary diarrhea, followed by two other group find that this channel have a physiological activity in two other cell type. So before going to discuss about the function, it's important to know where the channel is expressed. So this is the IF study. You can see these green signals is come from the apical pole of the acinar cells, not from the basolateral pole. So this channel is exclusively localized at the apical pole of the acinar cells, and this signal is not coming from the acinar ANO6 knockout gland. So this signal is specific. So this antibodies work well. So let's go for the in vivo study. For that one, I did the fecal chymotrypsin assay from the feces, from the mice feces. So when the food reaches to the duodenum, it stimulates CCK, secretin or acetylcholine secretion. The acetylcholine and CCK induce zymogen exocytosis, whereas secretin induce bicarbonate secretion. So the pancreatic juice come out from the pancreas into the duodenum, and one of the enzyme that still survive in the fecal matter, that is chymotrypsin, and clinician also use this chymotrypsin as an exocrine pancreatic assay. So I did measure the chymotrypsin in the fecal matter, and you can see the ANO6 knockout as mice have almost 80% reduced fecal chymotrypsin, that indicate these mice are suffering from the exocrine pancreatic disorder. So as you know, this chymotrypsin is stored in the zymogen granule. So if there is any zymogen granule distribution in this knockout mice, for that one I mark with the granules with the RAP27 that are mostly ready to fuse granules, and IP3 as a luminal membrane marker. You can see in the wild type the granules are present around the apical pole, but in knockout, most of the granules just stuck beneath the membrane. That indicate they have a problem in the exocytosis, because granules cannot fuse at the apical pole. Then I do the SEM to further characterize, and you can see in the knockout they have a high increased granular size as well as granular density that indicate intragranular fusion, and you can see the bigger granules that also indicate there is a maybe problem in the granular exocytosis. So then we move to the in vitro study for further characterize. To in vitro study, I take the pancreas, digest it and make the isolated acinar cells that behave similarly like whole gland, and stimulate with the CCH that increase the calcium through muscarinic pathway. In this condition, the calcium is only localized to the apical pole of the acinar cells, not going to the further. You can see the knockout are unable to respond to the stimulus, unable to secrete amylase. So still you can argue that they maybe have amylase synthesis problem, that's why they have lower protein, amylase protein, that's why they are unable to secrete amylase. To nullify that point, I measure the total amylase, and you can see there is no problem in the amylase synthesis in this knockout mice. So the problem is maybe come from the exocytosis. So what happens if I bypass the pathway, artificially increase the calcium? So incubate the isolated cells with the ionomycin that artificially increase the calcium without involving receptor, and I want to mention this calcium signaling not confined to the apical pole, but they spread through the cells and reach to the basal pole also. In this condition, the dose response curve shift towards the right, but the maximum exocytosis is conserved. So they have so in absence of ANO6, the exotic apparatus are unable to sense calcium. But in receptor dependent pathway, there is a change in the Vmax, the maximum exocytosis. So this leads to question: is there is any problem in the calcium oscillation? Because that is the trigger of the physiological exocytosis. Let's see what is calcium oscillation. So when I stimulate with the CCK, that induce the ER calcium release, and that further induce calcium entry through the ORI1 channel, and some of the calcium return back to the ER and efflux out from the cells. So this is known as store operated calcium entry and appear as an oscillatory pattern. You can see wild type have nice repetitive calcium, synchronous calcium oscillation. However, ANO6 knockout acinar cells have huge calcium influx, calcium in the cells. They have more calcium oscillation, but the pattern is very unsynchronous. So these have more calcium but unable to secrete anything. So because this high calcium is also a phenotype of the pancreatitis, so does this mice develop chronic pancreatitis? So to get the answer, I stimulate the cells, and you can see the wild type lose most of the granules, the Amazon granules, but in the knockout the granules are not secreted, but some of the granules start fusing at the basal pole. That also happen in the pancreatitis. When we go for the pathological stimulation, instead of the granules fusing at the apical pole, they start to fusing at the basal pole in absence of ANO6. That these granules also contain amylase, so that there is maybe chance of the increased serum amylase. So I look for the serum amylase, and these mice have a constitutive level of the serum amylase that is also indicator of the chronic pancreatitis. And then move for the histological study, and this knockout mice have severe inflammation. All this indicate these mice have developed the chronic pancreatitis. So what the possible explanations for this increased calcium but reduced exocytosis? I still don't have the answer of how these molecular things are happening. We are trying to figure it out. So we can say the in wild type, in presence of ANO6, this stimulate the luminal exocytosis but prevent basal exocytosis, and it also maybe prevent ORI1 channel to further entry of the calcium and prevent the further calcium overloading and cytotoxicity, and it may regulate the calcium sensitivity of the snare protein. We have to figure it out how they do when there is no calcium ANO6. there is an increased basolateral fusion, but inhibit luminal fusion and maybe calcium overloading. So, and this could be a therapeutic target for the exocrine pancreatic secretory disorder, like especially pancreatitis. So, future study in my lab, so how this exotic apparatus assembly and calcium regulation by the ANO6 and tie up, this is the most important thing I am now focusing on, the tie up the pH exposure with the exocytosis phenomena and find out if there is any mutation ANO6, because you can see there is a major regulator of the exocytosis. So, if there is any ANO6 mutation exists in the Indian core that drive the pancreatitis or secretory disorder. So, I acknowledge this is my former lab at the NIH, and he is my former lab. Dr. Malini was involved with me with this project, and this is my new workplace at Central University of Rajasthan with our lab student and my colleague. I would like to special thank to Professor Pramod Garg for his endorsement and Professor Sudipto Chaudhary for his invitation to the Pancreas Club, and I like to special thank to all of the members of the Pancreas Club to give me such platform to present my work. I'm happy to take the question.
Thank you so much, Dr. Jay. There's a question from the audience. Okay.
Very nice work. I like your cell biology. Nice images over there. More of a suggestion here. So your signaling is mostly calcium based. The data you've shown, ionomycin and the calcium signal itself. So we had published a paper long ago when I was in training in 2001 that there is a differential sensitivity of certain processes to cyclic AMP versus calcium based signaling. And yes, like secretin, for example, is more of a calcium-based secretion pathway. VIP is more of a cyclic AMP-based secretion pathway. Same with VIP. So and you had used carbachol over there in your first curve. It was carbachol. So the suggestion I would have is to kind of break this into compare the cyclic AMP-based signaling pathways, such as using cyclic AMP, dibutyryl cyclic AMP, or something that causes a non-calcium based signaling pathway, and compare and see is it a specifically calcium based phenomenon.
So go ahead then.
So that would be one clue as to, and the reason is because certain pathways such as actin based pathways are selectively calcium based. They are more resistant. They block calcium based secretion more than cyclic AMP based secretion. And yes, your protein localizes to the apical pole, and that's where all the actin regulatory secretory apparatus is also localized. So this could be something novel apart from phosphatidylserine, where your protein plays a role, such as through the actin regulatory pathway, which is only sensitive to calcium. So that's one suggestion. And then, do you have a response before I go to the next one?
Yeah. So there's a nice question. So I didn't show here. So I did work with the salivary gland. And the salivary gland, the exocytosis is completely cyclic AMP dependent. And we see the same phenotype. There is a blockage of the exocytosis, high cyclic AMP in the cells, but no exocytosis. Okay. So we are thinking that might be a problem with when there is the PS cannot expose out, that might block the fusion of the granules. That we are thinking.
Yeah, do it in acinar cells itself, pancreatic acinar cells, not parotid or salivary gland. So the other part is that you showed big blebs forming and granules going to the basolateral surface, am I right? So yes, that is pathological secretion which explains your high amylase at a baseline. However, it doesn't explain your first secretory curve. Your biphasic secretory curve to carbachol, which was higher in the wild type at even very high carbachol, but was lower in the knockout, even at a high carbachol concentration. So that conflicts with your observations of basolateral blebbing being the same, because if you're leaking basolaterally at a high concentration, that deficit in secretion should only be in the physiologic range, not in the supraphysiologic. The supraphysiologic should come back like you showed for ionomycin, it comes back at the high doses.
Yeah. So for the carbachol, so in the knockout, they are unable to secrete. So even they induce some of the, especially the physiological conditions. So
yeah, I'm talking supraphysiologic. After the peak, you are still higher in the wild type. If you go back to your first graph.
Yeah. Oh,
Yeah.
I got it.
Yeah.
Yeah.
Yeah, so after this peak, you see that the red line stays low. It is after the peak where you start making blebs and you start leaking basolaterally. And this remains down, so it doesn't catch up with that growth. So that's a bit of a conflict over there, that why did it not go up like that and come down? So it just stayed down all through. Am I making sense? So that whereas you see on the blebs, you are making a lot of blebs and you are going basolaterally. So you should have, eventually blebs will burst and secrete out, am I right? That's the high amylase you show in your mice.
Yeah.
So you reconcile that somewhere. You have to look at maybe a longer time point. Was this 30-minute secretion? How many secretions?
I think here it's like a 20-minute, 15
Yeah, so do a longer line. Maybe you'll see a blebs burst later on and rescue.
Okay. Yeah, that.